Hello, my name is Ryan Park, and this is my presentation for the Spring 2021's Research Symposium. And it is about the influence of living ends on polyethylene-based anion exchange membranes and their comparison to commercially produced AEMs. Now, to unravel and unpack everything I just said there, I wanted to start with a little bit of an introduction. So first, it is important to know what an anion exchange membrane is, or an AEM. And it is a semi-permeable membrane that selectively allows anions to pass through them while preventing other types of molecules from passing through. And the reason that they are important is because they are used in applications such as fuel cells, electrolyzers, redox flow batteries, reverse electrodialysis, and many other water purification applications. However, in many of these, the membranes are the limiting factor preventing the processes from becoming more economically or commercially viable. One of those things I said before was fuel cells, and so I have a little diagram here. And so you can see why they would be important. And from the diagram, you can see that hydroxide needs to be able to pass from the cathode to the anode side while preventing other molecules from also doing the same. And so the anion exchange membrane is what allows this to happen and for those reactions to continue. But as I said before, the membrane is what is limiting this process from becoming more feasible and viable. And the reason why our lab-produced membranes are special is because many other membranes end up degrading in aqueous or alkaline conditions, but our membranes show chemical and mechanical stability in these environments. And as I'll show later, our membranes have also showed to be able to function at a higher rate than some of the commercially used membranes today. Another important thing to know for this presentation is what the membranes we are using actually are. And so you can see here, this is a diagram of what our membrane looks like. It is a ABA triblock copolymer with this MPRD group attaching to this benzene ring. And so throughout this presentation, you'll hear me refer to three different membranes. One is membrane A, which is shown in this figure above. The other one will be membrane B, which is the same polymer except this disulfur end groups are lopped off. And then finally is Fumacep, which is a commercially produced anion exchange membrane, and so we'll be using that to compare to our membranes. So the first test I did was IEC, which stands for ion exchange capacity, and it essentially represents how positively charged the membranes are. And so if you can imagine, the more positively charged the membranes are, the easier it is for the negatively charged compounds to pass through them. And so here are the values for the corresponding membranes. As you can see, membrane A and B were both a fair amount higher than Fumacep. The next test was water uptake, which basically represents how much water the membranes take into their structure after being soaked. And so you can see from these graphs of time versus mass that after about the first hour or so, the membranes have taken up all about as much water as they will into their membrane as they would over the course of a much longer period. And so then taking the difference between this dry weight and the wet weight, you can see the water uptake value. And the values are recorded here, with membrane B having the largest swelling and Fumacep having significantly less swelling compared to our membranes. So the next major test that was done was trying to determine the diffusion rates of our compounds. And so what we did was we 3D printed this contraption here. And so when the test was ready, these two compartments would be screwed together with an O-ring to prevent spilling, and then we would have 0.1 molar NaNO3 on this side and 0.1 molar NaCl on this side. And then we would place the membrane right over that hole to prevent normal diffusion from happening. And so the only way the anions could pass through is through that membrane. And then we would measure the rate of chloride concentration changing over time in this cell. So the results of those diffusion tests is shown here in this graph, and as you can see in this graph, it is a graph of time versus chloride concentration, and over time the chloride concentration increases but at a decaying rate. And so this is the graph for Fumacep, membrane A, and membrane B respectively, just here to show that all the trials were reproducible and all similar to each other. So then taking the results from these tests and averaging them and plotting them onto one graph, it is much easier to see how they compared to each other. And so membrane A and membrane B were very similar, and so no real conclusion can really be drawn between the difference of these two. However, both performed significantly better than that of Fumasep, which is the commercially produced membrane, transferring nearly double the amount of mass in the same amount of time. This test was done again, this time with a 1 voltage potential difference applied between the two sides. And so once again we have Fumasep, membrane A, and membrane B, again just to show that we have similar and reproducible trials. Once again, averaging them and plotting them on the same graph, you can see this time membrane A performed better than membrane B. So it had a better increase in their difference between just diffusion and with the 1 voltage potential increase. However, both membrane and membrane B still performed significantly better than that of Fumasep. Looking specifically at membrane A, which is the one with living ends, or if you remember back to the polymer picture, membrane A is the one with that disulfur tail attached. And the difference between the diffusion and migration is shown here on this plot, where the migration was able to transfer about 10.5% more, had an increase of about 10.5% between the two trials. Membrane B, which is the one without that disulfur tail, essentially had no increase. And the conclusion that my research team and I have come to the conclusion of is that it's likely because membrane B has a much higher resistance, and so the electric field becomes smaller, and so the voltage difference becomes much less important, which is why the percent increase in this trial is probably as small as it is. And finally, for Fumasep, you can see that it had the largest increase of about 35%. However, if you remember back to the comparison, Fumasep also had significantly lower diffusion rates, which could account for why there's a larger increase between the difference in diffusion and migration. One of the more important values when it comes to membrane is the diffusivity. And so using the graphs that I showed you earlier and using Fick's law, I calculated the diffusivities. And so in order to use Fick's law, some important assumptions needed to be made, one of which is that over the time interval in which I applied it, it was at steady state. As you can see from the graph, it's never at steady state. However, if we consider a small time interval shown here, towards the end, the change in concentration is so small that a pseudo-steady state can probably be applied. Additionally, in order to use this equation, I assume that there were no boundary layers or resistance other than the membrane, and that equimolar counterfusion was occurring. And so, using known values such as volume, area, and the thickness of the membrane, along with calculated values such as the change in chloride concentration and the driving force at this time, I was able to calculate the diffusivities, which is shown here in the table along with the membrane thicknesses, which was an important number in calculating that value. Finally, I have all of the information put together in this one last final slide, as well as this graph, which is just a combination of all the graphs that you have seen before this. But similar conclusions can be drawn from it, just that membrane A performed the best in terms of mass transfer over time, especially with the voltage potential, and Fumasep performed the worst. And then comparing membrane A and membrane B, specifically against Fumasep, remembering that Fumasep is currently a commercially produced membrane and considered an industry standard right now, you can see that it not only performed better in the mass transfer, it had a higher IEC and a higher diffusion coefficient. The only downside is that our membranes had a significantly higher water uptake. However, depending on the application, this could not matter. And so overall, it would appear that our lab-produced membranes perform better than Fumasep. Then looking at specifically just the differences between membrane A and membrane B, remembering that the only difference is the living ends, you can see that membrane B had a higher IEC and water uptake, but a significantly lower migration increase and a lower diffusion coefficient. And so the differences between these two membranes suggest that the living ends are making a difference in both the characterization and performance between these two membranes. And then comparing the two, it would appear that membrane A is probably the better membrane in terms of application, just because of its better ability to transfer mass and its lower water uptake percentage. Additionally, because we can see that there is a difference between the two, it suggests that the living ends are the result between these, and the conclusion that we have drawn is that there's likely cross-linking occurring between those living end groups that is causing the chemical and structural differences between them. So that is my presentation, but before I end, I'd like to give a quick shout out to my graduate mentor, Ivy Wu, as well as the research professor I am working under, Dr. Andrew Herring. I hope you enjoyed, and thank you for your time.